Our third speaker is
Sure.
Dieter from Freiburg, Germany. His abstract is titled Spatial-temporal Remodeling of Tumor Architecture Under Therapy. Dieter, the floor is yours.
Thank you very much. Yeah, so it's really a pleasure to present and share some of our data, and I would just directly jump into the topic. So over the last years, my lab tries to understand the spatial architecture of glioblastoma. So we identified different niches that are basically show very high content of astrocyte microglia interactions. So we call these kind of immune-associated programs or other areas in the tumor that, for example, highly affected by metabolic alterations such as hypoxia. So to have this architecture in mind, we ask the questions how these architecture change when the tumor progress and during the time evolve into a recurrent tumor. The problem is that understanding the shift between de novo and recurrent tumors also contained a lot of different genetic drivers, epigenetic differentiation, transcriptional plasticity, and also a lot of the neural environment that we heard so far, and immunity processes that playing an important role to reshape the tumor into the recurrent or the relapse of these tumors. So to tackle these problems of a multidimensional problem, we designed a framework that we called Deep Sparta, containing multiple different deep learning architectures in which we now able to integrate multiple data types such as single cell sequencing data or especially spatial multiomic technologies. And this is just an overview of the spatial technologies that we're using. For proteomics, imaging mass spectrometry, then we have the Visium-based, so the array-based spatial transcriptomics approaches, along with metabolomics or in situ sequencing. So to ask now the question, what is the driver and what are the changes that basically define or differentiate a primary from a recurrent tumor? We designed a workflow in which we integrated whole Visium slides, so basically larger areas of the tumor, combined with in situ sequencing, in which we get a very high resolution, a cellular resolution on spatial transcriptomics, put these data together, and then use deep learning approaches to differentiate basically the temporal trajectory of these tumors. And by having these trained networks, we can then go and do in silico perturbations or simulation experiments to really module differences that appears from the shift from a primary to a recurrent tumor. So this is just a brief overview of the deep learning approaches that we use. We normally use graph attention networks, in which we here, for example, have a representation of the cellular architecture. We look at defined neighborhoods, so basically the neighbors of a center cell, and then it's three-order neighborhoods. And so basically each cell is then defined by its gene expression pattern, and each of these connection by the distance of these cells within the environment. So I don't want to step more in detail here in the architecture of the deep learning frame, but then we basically train the network to define and validate each of these neighborhoods, how likely this neighborhood comes from a primary or recurrent tumor. So to avoid additional bias variables, we then also train discriminators that basically define whether this network is not overfitting towards patient selection or specific histological defined regions. So basically having now a network that can perfectly validate each neighborhood and score each neighborhood, whether this comes from a primary or a recurrent tumor, we can then use tools which we basically summarize as explainable AI technologies to really look into the network and understand what are the decisions that were made by the deep learning approach to differentiate these different temporal stages of the tumor. So because of the sake of time, I would just go in the first approach, in which we specifically look into in silico simulation and perturbations of these cells. So just to give a brief overview, we trained our network, and then basically we can take a whole sample and then validate each neighborhood within the sample, whether basically this neighborhood is more likely to being derived from a primary or a recurrent tumor. So that means that also within a primary tumor, we can already identify regions that are more likely to look like recurrent tumor areas. So then we can zoom in these areas and look, for example, in the cellular neighborhoods that are very likely to be associated with a primary tumor architecture. So basically, these are examples of these cellular neighborhoods, and we can see that there's the most abundant cell types are tumor cells with like kind of a very ordered structure. So when we look on other areas that are more likely to look like recurrent tumors, we can already see that there's more stroma cells, there's also more macrophages, and in general, there's a higher entropy of these regions with a higher heterogeneity of cells. So having now just basically looking at the abundance, we can then jump in our simulation experiment and exchange, for example, cells or add some cells to these microenvironments and create simulated data. So basically we take an initial primary tumor and then we modulate the cells around, exchange or basically deposit the cells, and then feed them back into the neural network to ask whether this change that we have made in the simulation has any impact on the prediction being a primary or a recurrent tumor. So this is an example how it could look like of AC-like tumor cells, and then over 52 iterations, we changed these tumors into a mesenchymal-like tumor, in which we basically completely get rid of the astrocytic-like tumors. And then within these iterations, we can look whether, for example, here the epigenetic score change, and we see that the ATK2 phenotype that we have initially is then changing into the ATK1 phenotype when we enhance the mesenchymal component. But we can also do this now for the question whether this is a recurrent or a primary tumor. And we identify a lot of cells that basically are more likely to be enriched in these primary tumors, so in the de novo cancer. And then after a while, there is a couple of cells that's switching and showing higher abundance and higher likelihood to be enriched in these recurrent tumor microenvironments. And there is a couple of cells that are not really surprising, like CD8 T cells are enriched and also myeloid compartments. But one of the top candidates here were oligodendrocytes and neurons. So we looked there a little bit more in detail. And so this is an example of one simulation in which we basically take a primary tumor and then add more oligodendrocytes, in which we can see that the recurrence score is rising. We can also do this for therapeutic approaches and identify, for example, B cells highly to be enriched in environments after PD-1 inhibition, which can then also be confirmed in single cell data and high resolution spatial transcriptomic data. So basically the problem is very complex, the shift between a primary and recurrent tumors, but we can use modern deep learning architectures to really understand what is happening in the microenvironment, also use simulation and other explainable AI tools to really understand how this change happens. So with this, I want to thank and happy to take some questions.